So what if I told you that a single microscopic enzyme could completely change the clarity of your morning tea, help produce life-saving antibiotics, clean up toxic waste, and basically unlock a massive web of industrial biotechnology? Pretty wild, right? Well, welcome to This Explainer. Today, we're unpacking the molecular magic behind one of the most incredibly versatile biological tools on the planet, the Tannase enzyme. We're going to dive right into the mechanics of this absolute microbiological powerhouse. And that question, it isn't just some fun hypothetical. It's actually being answered right now, as we speak, in bioreactors and laboratories all around the world. We're literally connecting the dots between hardcore biochemistry and global sustainability. So today, we're going to explore exactly how this incredible biological catalyst works and the profound real-world implications it has across, honestly, just a massive number of industries. To wrap our heads around this, we'll follow a pretty clear path. First, we'll break down the Tannase enzyme solution itself, then we'll check out its microbial sources. After that, we'll look at production and properties, before finally scaling all the way up to its huge industrial and medical applications. All right, part one, the Tannase enzyme solution. Let's zoom right into the micro level to figure out what tannins and tannase actually are. Okay, so to really understand tannase, we first got to understand tannins. Tannins are secondary metabolites found in plants. Think of the bark on trees, leaves, or Chinese gulls. They are absolutely notorious for forming complexes with proteins, which basically means they can bind to and denature them. Now, check out the specific definition of tannase, also known as tannin acyl hydrolase. This is the precise enzyme that breaks down those really tough, complex tannin molecules. You can think of it like an elegant set of biochemical scissors. It specifically targets and hydrolyzes the ester and dipeptide bonds to neutralize the tannin, yielding some incredibly valuable byproducts in the process. Let's walk through the exact biochemical steps of this hydrolysis process, because it's actually pretty fascinating. First, the Tannase enzyme zeroes right in on hydrolyzable tannins, like tannic acid, which is its natural substrate. Second, it rolls up its sleeves and gets to work cleaving those ester and dipeptide bonds. And finally, step 3, and this is the really crucial part, it releases gallic acid and glucose as the final products. And gallic acid? It is definitely not just a waste product. As we'll see in a minute, it's a highly sought-after molecule in pharmaceuticals and fine chemicals. So Tannase essentially takes a stubborn plant defense mechanism and turns it into absolute molecular gold. So now that we know how it works, where do we actually find this stuff? That brings us to section 2, microbial sources of tannase. While you can actually find tannase in tannin-rich plants, and even naturally in the digestive tracts of cattle, the biological factories of choice for modern biotech are, without a doubt, microbes. We're talking bacteria, fungi, and yeast. They all offer distinct, amazing sources for extraction, and microorganisms are heavily favored over plant or animal sources for a few key reasons. For one, microbial enzymes are vastly more stable. Plus, from a purely industrial standpoint, microbes can be cultured in massive quantities super fast, and you can subject them to genetic manipulation far more easily than animals or plants. We're essentially just harnessing the incredible biochemical diversity of the microscopic world. And honestly, this perfectly brings us to the real star of the show here, the Aspergillus genus, and specifically Aspergillus niger. This mold is an absolute microbial workhorse. Now get this. Tannic acid in high concentrations is highly toxic to most organisms, right? Because it denatures our proteins, just like we talked about. But Aspergillus niger, it has this incredible, resilient saprophytic lifestyle. It can tolerate tannic acid concentrations up to an astonishing 20% without any negative effects on its growth or enzyme production. Actually, scratch that. It doesn't just tolerate it, it thrives. It uses the tannic acid as a sole carbon source, just pumping out massive amounts of tannase, to break it down. It's a survival mechanism that we get to capitalize on. All right, so we've got our microbial superstar, but how do we actually manufacture the enzyme efficiently? Let's get into section 3, production and properties. When it comes to industrial scale production, researchers usually weigh two main methods against each other. First, you have submerged fermentation. This involves growing microorganisms in a liquid nutrient medium. It's kind of the classical, traditional approach. But then you have solid-state fermentation, or SSF, which involves growing the microbes on water-insoluble substrates with very, very little free water. And the difference between the two is staggering. SSF often blows the other method out of the water for Tannase. Why? Because it yields up to 5.5 times more enzyme than submerged fermentation. I mean, that's a huge difference. Plus, the enzymes produced via SSF are typically extracellular, and they show way broader stability across all sorts of different temperatures and pH levels. These lab methods directly dictate the physical conditions our Tannase needs to fire on all cylinders. The big takeaway here is that Tannase loves a good acidic environment, usually peaking right between a pH of 4.3 and 6.5. Its ideal temperature range is a pretty comfortable 30 to 40 degrees Celsius, though, some specialized strains can operate much higher. But look at those bottom 2 points on the properties list. This is just fascinating regulatory biochemistry. The presence of specific metal ions completely changes the enzyme's efficiency. Magnesium and manganese, they act as activators. They kick the enzyme right into high gear. But throw in some iron or cobalt and they can severely inhibit or literally completely stop its activity. So we've covered the microbiology, we've looked at the lab conditions. Now let's pick up the pace and scale this all the way up to the macro level with Section 4, Industrial and Medical Applications. When you map it all out, the industrial web created by this single enzyme is just mind-blowing. Tannase isn't just some niche chemical tucked away in a lab somewhere. It sits right at the center of an incredible network that connects totally disparate global industries. From the food we eat and drink to the clothes we wear to the medicines that literally save our lives, Tannase is a critical catalyst. I mean, we're talking pharmaceutical production, the dye industry, photography, cosmeceuticals, leather processing, even biofuel production. It is the perfect example of how mastering just one single biological mechanism can have these massive, sprawling ripple effects across all of human technology. Let's anchor all that biochemistry to some tangible things you probably see every day. In the food and beverage world, Tannase is the hidden secret behind clear fruit juices and wines. It actually removes the high tannin content that causes all that haze, sedimentation, and bitterness. It's also the exact reason your instant tea dissolves so easily in cold water. Over in pharmacology, remember that gallic acid we talked about earlier? Well, it's a primary precursor for manufacturing trimethoprim, which is a vital, broad-spectrum antibiotic. Environmentally, Tannase is a total hero in bioremediation. It breaks down the toxic tannin-rich effluents pumped out by leather tanneries long before they can poison local waterways. And out in the agricultural sector, treating animal feed with Tannase reduces the anti-nutritional effects of tannins, which vastly improves how well livestock can actually digest their food. It's a total game changer. It really is a miracle enzyme seamlessly bringing together biotechnology, medicine, industry, and sustainability. And that brings us to our final thought for today. As we face some pretty huge global challenges in sustainable manufacturing, waste management, and medicine, how will green enzyme technology, just like Tannase, continue to shape the future of sustainable biotechnology? It is such a fascinating space to watch. If you're into biotechnology, medical science, and the innovation shaping our future, be sure to like, share, and subscribe for more STEM and science content. Thanks so much for joining me on this explainer, and as always, keep learning.